Welcome back to the deep dive. Today we are setting aside the gardening tools and, well, we're picking up the magnifying glass, I guess.
That is a very specific and slightly ominous way to start the show.
Right, well, think about it. When we looked at bacteriology in our previous research, it felt a lot like agriculture to me.
Yeah, planting seeds on a petri dish.
Exactly. You plant a sample on the agar, you give it some nutrients, maybe a little warmth, and you just wait for a garden to grow. It's distinct, it's visible, and in a weird way, it's comfortable.
And today is not comfortable.
Today feels like forensic science. We are dealing with viruses, and these things do not grow on their own.
No, they don't.
They are obligate intracellular parasites. They're invisible hijackers. You cannot just smear them on a plate and watch them bloom like a bed of roses.
And that is the fundamental disconnect that students often struggle with. I mean, with a bacteria, you are looking for the organism itself sitting right there on the agar.
Right.
But with a virus, you are almost never seeing the perpetrator directly. You're looking for fingerprints.
Fingerprints, broken windows, maybe a dropped wallet, or a note left behind.
Exactly. We're hunting for their genetic code, the DNA or RNA fingerprints, or we're looking for the damage they leave behind in cells.
The broken windows.
Right, the broken windows, which we call cytopathic effects. Or we're looking for the body's security response, which would be the antibodies.
Detection without growth. That's the theme of the stack of papers we have today. We are breaking down Chapter 39 of Murray's Medical Microbiology with some heavy tactical support from Jowett's Chapter 47.
It's a great combination of sources.
Yeah. And the mission today is to take you from that moment at the bedside swab in hand all the way into the molecular machinery of the lab.
And for the medical students or professionals listening, this is really high-yield territory.
Absolutely.
We aren't just talking about abstract biology here. We're talking about why you don't freeze a flu swab, why owl's eyes matter on a pathology report, and why a negative rapid test does not always let you off the hook.
Let's start at the very beginning, the crime scene. You're standing next to the patient, you have a swab. What is the golden rule of viral specimen collection?
The rule is simple. Timing is everything. You have to strike while the iron is hot, or rather while the virus is actually shedding.
So we're talking about the acute phase.
Right. Viral shedding is often incredibly transient. If you're dealing with a respiratory virus, flu, RSV, COVID, that window where the virus is actively replicating and being expelled is tight.
Like what, 3 to 7 days?
Usually 3 to 7 days, yes.
So if a patient walks in 2 weeks after their cough started and says, "Doc, figure out what this was," you're in trouble.
You're way too late for isolation. The virus is likely gone. At that point, you're not looking for the burglar anymore, you're looking for the police report.
Which means serology.
Exactly. You are stuck with serology looking for antibodies, which is a retrospective diagnosis. It tells you history, not current events.
Okay, so we catch them early in the acute phase. But here is the part that I think really trips people up. Viruses are not just tiny diphtheria.
Far from it.
They have a completely different physical structure, and that dictates how we handle the sample. I was reading about the distinction between enveloped and non-enveloped viruses in Murray. It feels like the difference between carrying a soap bubble and carrying a golf ball.
That is a perfect analogy.
Yeah.
Let's look at the enveloped viruses first.
Okay.
Things like influenza, herpes, simplex, varicella. That envelope is just a lipid membrane. It's a biological soap bubble derived from the host cell.
And soap bubbles are notoriously fragile.
Incredibly fragile. If they dry out, they pop. If the pH shifts, they pop. And here's the kicker that shows up on board exams constantly.
Oh, let me guess. Freezing.
Yes. If you freeze them incorrectly, they are completely destroyed.
Wait, but I thought cold was good. We put milk in the fridge. We put meat in the freezer to keep it safe.
Cold is good. Freezing is dangerous. If you put an enveloped virus in a standard household or clinic freezer, which is usually around minus 20 degrees Celsius, you are killing it.
Why is minus 20 the magic kill zone?
It's all about the physics of crystallization. At minus 20, water forms large, jagged ice crystals. Those crystals act like microscopic daggers.
And they just shred the bubble.
They puncture that lipid envelope. You essentially shred the virus.
So you have got a sample of herpes simplex, you toss it in the standard clinic freezer overnight, and the next day, the lab results come back negative. Not because the patient is clear, but because you literally destroyed the evidence.
Precisely. It is a false negative caused entirely by bad handling.
So what's the alternative?
If you need to transport it, use wet ice. Keep it at 4 degrees Celsius. That keeps it stable without freezing.
And for long term?
If you need to store it long term, you have to go deep, -60, -70 degrees. You need a deep freeze that freezes the sample so instantly it bypasses that jagged crystal formation phase entirely.
That makes a lot of sense. Now contrast that with the golf balls, the non-enveloped viruses.
Right, these are your adenoviruses, your enteroviruses. They have a rigid protein capsid.
So no lipid bubble.
No lipid bubble. They are tough. They can survive on door handles, they can survive in stomach acid, and they can handle temperature fluctuations much, much better. They're basically the tanks of the viral world.
But regardless of whether it's a bubble or a golf ball, we don't just drop the swab in a dry tube, right? We use VTM, viral transport media.
Right. And this confuses people who are used to bacteriology. In bacteriology, transport media often has nutrients to help the bugs grow.
Because you want more of them.
Exactly. But in virology, VTM is a life support system. It is a stasis field. We aren't trying to grow them yet.
We just want to stop them from falling apart.
Right. The media usually has proteins like gelatin or serum albumin to stabilize that envelope. Jadwetz also mentioned specific sucrose and phosphate buffers to maintain the perfect pH. But it also has something else that is absolutely crucial.
Antibiotics.
Yes, antibiotics.
Which sounds completely counterintuitive when you first hear it. We are looking for a virus. Antibiotics killed malaria. Why would we add something that kills the wrong thing?
Think about where you're swabbing. The throat, the nose, even a skin lesion.
Dirty places.
Very dirty places. They are teeming with normal flora bacteria. If you don't put antibiotics in the transport tube by the time that sample gets to the lab, the bacteria will have overgrown everything.
Oh, because they replicate so much faster.
Exactly. They will eat all the nutrients, they'll change the pH, and they will release toxins. They will ruin the cell culture you're eventually going to try to use to grow the virus.
So the antibiotics are basically the bouncers. They clear out the riffraff so the VIP, the virus, can actually be seen.
That's it. You are suppressing the background noise so you can hear the signal.
Okay, so the sample survives the trip. It arrives at the lab. Now, before we even try to grow it or sequence it, we can sometimes just look at the damage. This brings us to cytology, the broken window stevia we mentioned.
This is the fastest, cheapest, and oldest way to diagnose. We're looking for CPE, cytopathologic effects.
Meaning the virus hijacks the cell and the cell changes shape. It starts to look weird.
And there are specific kinds of weird that are practically diagnostic signatures.
The text highlights 3 big patterns that you need to know. Let's walk through them for you listening. First up is syncytia.
Syncytia. It comes from Greek, meaning cells together.
The fused cell.
Right. Some viruses, specifically the paramyxoviruses like measles and RSV, but also herpes and HIV, produce a fusion protein.
And what does that do?
When they infect a cell, they dissolve the borders between that cell and its neighbors.
So the walls literally come down.
And the contents merge. You end up with one giant massive cell with dozens of nuclei floating around inside it.
It looks like a bag of marbles.
Or a cluster of grapes. If you look under a microscope and see these multinucleated giant cells, you're dealing with a fusion-inducing virus.
I've heard this mentioned in the context of the Zenk smear.
Yes, a classic bedside test. If a patient has a vesicle, a blister, maybe it's herpes, maybe it's varicella, you unroof the blister, scrape the bottom, stain it, and look for those giant syncytial cells.
Quick, right?
Very quick, though. Not as sensitive as modern molecular methods, but historically very important.
Okay, pattern number 2 for cytopathologic effects. Inclusion bodies.
If syncytia are the broken walls, inclusion bodies are the piles of trash or the factories left behind inside the cell.
So they're aggregates of viral proteins or replication sites.
Yes, and where they are located tells you a lot about who the culprit is.
Right, because DNA viruses usually replicate in the nucleus, so you find the inclusions there. RNA viruses usually replicate in the cytoplasm, so you find them out there.
Generally speaking, yes. That's a great rule of thumb for exams.
The most famous one has to be the owl's eye.
Oh, it's impossible to go once you see it. This is cytomegalovirus, CMV.
Which is a herpes virus.
Right, so it replicates in the nucleus. It creates this massive, dark, basophilic blob right in the center of the nucleus, and it pushes the normal chromatin all the way to the edges, creating a clear halo.
It literally looks like an eye staring back at you from inside the cell. It is honestly haunting.
It really does. And if you see that in a lung biopsy or urine sediment, it is CMV. Done. That's a huge textbook keyword.
When you have Cowdery type A inclusions.
Also intranuclear, but they look different. They are eosinophilic, meaning they stain pink. That is the hallmark of herpes simplex and varicella zoster.
And what about the cytoplasmic ones, the ones outside the nucleus?
The classic example you have to know is the Negri body. This is pathognomonic for rabies.
Rabies is an RNA virus, right?
Yes. It sets up shop in the cytoplasm of brain cells. Specifically, you want to look at the Purkinje cells in the cerebellum or cells in the hippocampus. You see these pink eosinophilic blobs floating in the cell body.
It's morbidly fascinating. You are looking at the physical machinery of the infection in someone's brain tissue.
You are. But, you can only see so much with a light microscope. Sometimes you need to see the virus itself.
And for that, you need the electron microscope, the dinosaur of the lab.
It is a bit of a relic for routine clinical diagnosis these days. It's expensive, it's technically very difficult, and worst of all, it's highly insensitive.
How insensitive.
You need millions of viral particles per milliliter just to find one on the screen. It's like trying to find a specific grain of sand on a beach.
So why do we even mention it? Why is it still taking up space in Murray Chapter 39?
Because of poop.
Poop, go on.
Specifically rotavirus. Rotavirus is a major cause of severe diarrhea in kids. When a child has rotavirus, they shed it in stool in absolutely massive quantities. We're talking 10 to the power of 10 particles per gram of stool.
Wow, so the haystack is basically made of needles at that point.
Exactly. And rotavirus has a very distinct shape. It looks like a wheel. Rota means wheel, so you can actually see it pretty clearly.
And Jawitz talks about a trick to make it even easier, right? Immunoelectron microscopy.
Yes. You mix the stool sample with antibodies that specifically bind to the rotavirus. The antibodies act like magnets, clumping the individual viruses together into big, visible rafts. It makes them much easier to spot under the EM.
Okay, so EM is mostly for history books, research, and pediatric stool samples. Now let's get to the part that used to be the gold standard before the genetic revolution took over. Viral isolation, actually growing the beast.
This is where we try to replicate the crime in a controlled environment. But remember, viruses need living cells. They won't grow on agar. So we have to use cell cultures.
In reading through Murray, it seems like not all cell cultures are created equal. There's a hierarchy of immortality, if you will.
That is a dramatic but accurate way to put it. We have 3 tiers of cell lines. Tier 1 is primary cell cultures.
These are the fresh ones.
Right. You take an organ historically, usually a monkey kidney. You dissociate the tissue, chop it up, and put the cells in a tube. These are normal, healthy cells.
And what are they good for?
They're great for growing picky viruses like influenza or parainfluenza. But because they are normal cells, they have a biological destiny. They age.
They undergo senescence.
Exactly. They divide a few times and then they die. Their lifespan is very limited.
So they're high maintenance. Then we move to tier 2, diploid cell lines.
These are usually derived from human fetal tissues. They're a bit more robust than primary cells. They can last for about 50 divisions before they undergo senescence. We use these for a broad range of things like HSV and CMV.
And finally, tier 3, the zombies, the continuous cell lines.
The immortal ones. These are tumor cells. The most famous, of course, are HeLa cells taken from Henrietta Lacks cervical cancer back in the 1950s, or HEP2 cells.
And because they are cancer, they've lost the stop signal.
Right. They will divide forever as long as you keep feeding them.
That sounds perfect. I mean, why do we not just use immortal cells for everything?
Trade-offs. They are easy to keep alive, but they are not always the best hosts. Some viruses just don't like to infect them.
They're picky eaters.
Yeah, they're less sensitive for things like flu. But for adenovirus or RSV continuous lines are the absolute workhorses of the lab. Now, there is a major logistical problem with growing viruses. It is slow. A bacterial plate might be ready overnight. A viral culture for something like cytomegalovirus can take weeks.
And if you are a transplant patient with CMV pneumonia, you don't have weeks.
You'd be dead before the result came back.
Exactly. That was the major limitation of clinical virology for decades. But then we got clever. Jawitz describes a fantastic technique called the shell vial culture.
This is the need for speed hack.
It is. So normally you place the liquid sample on the cells and wait for the virus to naturally drift down, attach, and infect them. It takes forever. So instead, we force the issue.
How do you force a virus?
Centrifugation. We put the sample in a small vial that has a cover slip of cells at the bottom, and we spin it. We spin it at 700 times gravity.
You are physically smashing the virus against the cell door.
We are breaching the perimeter. By physically forcing the virus onto the cells, we skip the entire waiting period for attachment and entry.
That's brilliant. And then, instead of waiting for the cells to die and show those cytopathic effects, which again takes weeks, we just wait 24 to 48 hours. The virus gets in, starts making its early proteins, and then we stop the clock.
And you look for those proteins.
Right, we stain the cells with fluorescent antibodies to see if those early viral antigens are present.
It's like checking the security footage after 1 hour instead of waiting for the bank vault to be totally empty.
Exactly. It cut the diagnosis time for CMV from several weeks to roughly 1 or 2 days. It was an absolute revolution for patient care.
That brings up a good point. We've been talking about growing the virus, but how do you actually know it's there? We mentioned CPE, the cells looking sick. But are there other ways to know a culture is positive if the cells haven't lasted yet?
There are. Sometimes the cells look perfectly healthy under the microscope, but they are actually teeming with virus. One way to catch them is hemadsorption.
Hemadsorption. This sounds like the sticky test.
It relies on a specific trick used by influenza parainfluenza and mumps. When these particular viruses infect a cell, they force the host cell to display a viral protein called hemagglutinin on its surface.
So the infected cell sprouts these sticky spikes?
Right. So if you pour a solution of red blood cells, usually guinea pig RBCs, into the culture tube, those RBCs will get snagged on the spikes. They will stick directly to the surface of the infected cells.
So under the scope, it looks like the cell is covered in berries.
Yes. And that is hemadsorption. It finds the factory, it finds the infected cell.
And the textbook makes a point to distinguish this from hemagglutination.
Yes, a very important distinction. Hemadsorption is red blood cells sticking to the infected cell monolayer. Hemagglutination is free-floating virus in the liquid clumping the red blood cells together into a web.
I see. So hemadsorption equals cells sticking to cells. Hemagglutination equals free virus sticking to cells.
You got it.
Now, there was one more detection method in culture that I thought was just brilliant genetic engineering, ELVIS.
The enzyme-linked virus-inducible system. And sadly, no, it has nothing to do with rock and roll.
Such a missed marketing opportunity.
Truly. But the science is incredibly cool. You take a cell line and you implant a gene from E. coli, the LAC-Z gene. This gene produces an enzyme called beta-galactosidase, which turns a specific chemical substrate blue.
But here's the trick, right? You hook that E. coli gene up to a viral promoter.
Exactly. A promoter is a genetic switch. In this case, it's a switch that only a virus can flip.
Under normal conditions, the gene is silent. The cell is colorless.
Right. But if herpes simplex enters the cell, it naturally tries to turn on its own genes. In doing so, it activates that specific promoter we implanted. The switch flips, the LAC-Z gene turns on, the cell makes the enzyme, you add the substrate, and boom.
The infected cells turn bright blue.
Yes. So you don't need a PhD in microscopy to look for subtle changes in cell shape or Cowdrey type A inclusions. You just look at the well and say, are there blue spots?
It removes all the subjectivity. It's binary, blue or not blue.
Exactly. Very fast and very specific for HSV.
Okay, so we've grown it, we've detected it. But sometimes, especially in research or vaccine work, we don't just need to know it's there. We need to count it. We need to know the viral load or the potency of a sample. This leads us to viral quantitation.
This gets a little math heavy in the text, but the core concepts are key. We talk about titers, and you will see terms on exams like TCD50 or LD50.
50% endpoint.
It's a statistical measure. TCD50 is the tissue culture dose, where 50% of your inoculated cell cultures show CPE and die.
And LD50.
That's the lethal dose, where 50% of your test animals die. And ID50 is the infectious dose, where 50% show symptoms or produce antibodies. It's a way of standardizing how strong is this specific batch of virus.
And then there's the plaque assay. This feels the most like the bacteriology counting colonies method we talked about in earlier deep dives.
It is the exact viral equivalent. You grow a lawn of cells, a nice flat carpet of cells in a dish. If a single infectious virus lands on one cell, it infects it, it replicates, kills it, and the progeny spread to the immediate neighboring cells.
And it creates a dead spot, a hole in the carpet.
Yes. We call that clear zone a plaque.
So one plaque equals one infectious virus particle.
Ideally, yes. But here is a fascinating nugget from Murray that really changes how you think about viral replication. The particle to PFU ratio.
PFU meaning plaque forming unit.
Right. If you take a sample and look at it under an electron microscope, you might see 1,000 physical viral particles. You can count them. But if you put that exact same sample on a plaque assay, you might only see 10 plaques form.
So 990 of them were deads.
Exactly. Viruses are incredibly sloppy manufacturers. They replicate so fast, they make a lot of junk. Empty capsids, broken genomes, missing envelope proteins. Just because it looks like a virus under a microscope doesn't mean it actually works.
That is a huge distinction for people to understand. Physical presence does not equal infectivity.
A critical concept to hold on to, especially when we start talking about molecular detection.
Which is the perfect transition. Let's leave the petri dishes and the cell cultures in the past. We're moving into the modern era of the lab. The techniques that have basically taken over the world in the last 20 years. The detection of viral genetic material.
The molecular revolution. This shifted the entire paradigm from growing to finding. We stopped waiting for the virus to replicate itself and started hunting for its genetic code.
Because it's faster.
It's much faster. It's safer because you aren't growing gallons of dangerous pathogen in the lab, and it is infinitely more sensitive.
And the absolute king of this hill is PCR.
The polymerase chain reaction. The molecular photocopier.
Most people know the acronym now, especially after the last few years. But functionally, what does it do? It amplifies a very specific, tiny segment of DNA. It turns a needle in a haystack into a haystack made entirely of needles.
But there is a catch. Standard PCR only works on DNA. And some of our worst clinical enemies, influenza, HIV, SARS-CoV-2, are RNA viruses.
So how do we photocopy RNA?
You need a translator. You need an extra step at the very beginning called reverse transcription. You use an enzyme called reverse transcriptase to turn the viral RNA into complementary DNA or cDNA.
And once you have the DNA, then you can run the photocopier.
Exactly. That is RT-PCR, reverse transcriptase PCR. It's the bedrock of modern virology.
And then we have real-time PCR, or qPCR. This isn't just about finding the virus, it's about counting it as it happens.
Right. This is the absolute standard of care for chronic viral management like HIV and hepatitis C. Real-time PCR measures the accumulation of DNA glow-in-the-dark style using fluorescent probes.
So the more virus you started with in the patient's blood, the faster that fluorescent signal gets bright.
Exactly. And that gives us the quantitative viral load.
Why is that number so clinically important to track?
Because it is the fuel gauge for the infection. If I start a patient on antiretroviral drugs for HIV, I need to know if those specific drugs are working. I want to see that viral load drop from 1 million copies down to undetectable.
And if it spikes back up?
If it spikes back up, it tells me the virus is mutated and become resistant to the drugs or the patients stop taking their pills. It completely guides the therapy in real time.
The text also emphasizes these multiplex assays, the syndromic panels.
A total game changer for the emergency room. Before you had to guess, a patient comes in coughing. Is this flu? Is it RSV? Is it adenovirus? And you'd have to order separate tests for each.
Which takes time and money.
Yes. Now with multiplex PCR, you take one single swab, run it on a respiratory panel, and it checks for 17 or 20 different viral and bacterial targets simultaneously.
It tests for the syndrome, not the specific bug.
Exactly. It catches things you wouldn't have even thought to order. It takes the guesswork out of the initial triage.
Now, there is one more molecular technique mentioned in Chapter 39 that I want to touch on. In situ hybridization, or 5SH, this involves looking at actual tissue again.
Think of this as a molecular map. You take a physical biopsy, say, of a cervix or lung tissue. You treat it with a DNA probe that perfectly matches the genetic sequence of the virus you suspect, say, HPV. And that probe has a fluorescent tag attached to it.
So if the virus is there, the probe binds and it glows under a specific light.
But critically, it glows inside the preserved tissue architecture. It proves that the virus is actually inside those specific damaged cells, causing the dysplasia or the problem rather than just floating nearby in the mucus.
It connects the criminal directly to the crime scene.
Exactly. Very important for viral pathology.
So we've looked for the whole virus via culture. We've looked for the genetic code via PCR. Now let's quickly touch on the middle ground, detection of viral proteins.
This is usually antigen detection. We use known antibodies to essentially sandwich the viral proteins and make them visible.
These are your rapid tests, that flu swab at the urgent care.
Yes. The big pro is that they are fast, 15 minutes point of care. But the con is much less sensitive than PCR. You need a relatively massive amount of viral antigen in the swab to trigger a positive result.
So a negative rapid test does not always mean you are clear.
No, it just means you don't have a huge amount of virus actively shedding at that exact moment. That is why we often confirm negative rapid tests with a PCR if the patient looks really, really sick.
Okay, finally we come to the last major section of the chapter, serology, the history book of the immune system.
This is fundamentally different from everything else we have discussed today. In culture and PCR, we are looking for the virus itself. In serology, we are looking for the host. We're checking the body's reaction logs.
We are measuring antibodies. And the specific type of antibody tells us the timeline of the infection. We have two main characters here that students need to know: cold, IgM, and IgG.
I always like to think of IgM as the Marines.
The first responders.
Exactly. They hit the beach first. IgM appears very early in the acute phase of the infection. It is a massive molecule. Structurally, it's a pentamer, so 5 individual units stuck together in a star shape.
Which makes it great at grabbing and clumping a lot of virus quickly.
Yes, it's great at agglutination. But it doesn't stay around long. IgM levels usually peak and then disappear within a few weeks or months.
So if you draw blood and find IgM against a virus, it means the war is happening right now, or it literally just finished.
Correct. IgM equals acute or recent infection.
And then comes IgG.
The National Guard. They arrive a bit later to the fight, but they garrison the town. They stick around for years, often for the rest of your life.
So IgG indicates past exposure or successful vaccination.
Exactly. But here is the major clinical catch that trips people up. What if you want to diagnose an active ongoing infection, but the patient only has IgG? Maybe the IgM window has passed or the test isn't great.
A single high level of IgG tells you nothing about today. It just says at some point in your life you saw this virus.
Right. So how do we prove the illness happening right now is caused by that specific virus using only IgG?
You need the golden rule of seroconversion. You need paired sera.
Yes, 2 separate blood samples.
One taken right now when they are sick, the acute sample.
And one taken 2 to 3 weeks later when they are recovering, the convalescent sample. You compare the antibody levels or the titers between the two.
And we're looking for a very specific math outcome, the 4-fold rise. Let's explain this because it's so critical.
Okay, so titers are measured in doubling dilutions, 1 to 2, then 1 to 4, then 1 to 8, then 1 to 16, and so on. To definitively prove an active infection during that specific time window, you need to see a jump of at least 2 doubling steps between the acute and convalescent sera.
So going from a titer of 1 to 8 in the first sample up to 1 to 32 in the second sample?
Exactly. That is a 4-fold increase. It proves the immune system was actively ramping up production of those antibodies during those exact 3 weeks.
What if it just goes from 1 to 8 up to 1 to 16?
That's only a 2-fold rise. Not enough. That could simply be lab error or normal biological noise. You need that decisive 4-fold spike to say, yes, this virus was the cause of the recent illness.
But serology has limitations, right? It's slow.
It is entirely retrospective. By the time you get that second convalescent sample 3 weeks later, the patient is often already fully recovered, or unfortunately, they've passed away.
So you can't use it to decide what drug to give them on day 1?
No. It's more useful for epidemiology population studies or for difficult retrospective diagnoses where the virus just wouldn't grow in culture.
And there are issues with cross-reactivity, too.
Yes. The immune system is not always a precision instrument. The antibody your body made against parainfluenza might structurally look a lot like the antibody against mumps. So a mumps serology test might falsely read positive.
There's also the issue of immune complexes binding up the antibodies, like in hepatitis B.
Right. In hepatitis B, the virus produces so much surface antigen that it acts like a sponge. It binds up all the free antibody in the blood. So when you run a test looking for that antibody, it comes back negative. Not because the patient didn't make it, but because it's all tied up in complexes.
Fascinating. We have covered a massive amount of ground today. From ice crystals destroying lipid envelopes to counting plaques in a lawn of immortal cells. Let's try to bring it all together.
If you boil it down, we really moved through a diagnostic evolution. We started with the visible, seeing the cytopathic effects like syncytia or inclusions. Then we moved to the biological, actually growing the virus in cell culture. Then to the molecular, finding the genetic code with PCR. And finally to the historical checking, the host antibodies with serology.
And for a quick mental check for you listening out there, if you need it fast.
You go for an antigen test or rapid PCR.
If you need to know exactly how much virus is there.
You know, real-time PCR to get a viral load.
And if the virus just will not grow in culture at all.
The serology is your backup. Look for the footprints.
Now here is a provocative thought to leave you with before our review.
Yeah.
We talked about those multiplex panels. Testing for 20 viruses at once with incredible molecular sensitivity. We're getting really, really good at finding viral DNA. But maybe we're too good.
That is the big modern debate. Just because you find a trace of viral DNA in a patient's nose, does it actually mean that specific virus is causing their symptoms today?
Right, because we shed viruses for weeks.
Exactly. You might have a rhinovirus trace lingering from a cold you had last month, but your severe symptoms today are actually bacterial pneumonia. With such hypersensitive tests, We risk overdiagnosing the innocent bystander virus and missing the true culprit.
Detection does not automatically equal disease. Interpretation requires clinical context. It requires a doctor looking at the patient, not just a machine reading a barcode.
Context is always king in microbiology.
All right, before we sign off, let's lock this knowledge in with some rapid recall, single best answer style. I am going to ask a question. I'll pause for a second for you to think about it, and then we will give the answer and the why. Ready?
Ready.
Question 1. A sample for viral isolation is delayed in transport. A well-meaning nurse places it in a standard clinic freezer at -20 degrees Celsius overnight. Which type of virus is most likely to be completely destroyed by this action? Think about it for a second.
The answer is an enveloped virus, like influenza HSV or HIV. The large jagged ice crystals that form at -20 degrees physically puncture and disrupt the fragile lipid envelope, destroying infectivity. If you need to review this, check the specimen collection section of Chapter 39.
Question 2. You perform a Zank smear on a scraping from a patient's skin vesicle. Under the microscope, you observe large multinucleated giant cells. Which mechanism of viral replication directly causes this cytopathologic effect?
The answer is cell fusion, or syncytia formation. Viruses like HSV, VZV, and paramyxoviruses express fusion proteins that dissolve cell membranes, merging adjacent cells into one giant mass. Review the cytology and CPE section for more on this.
Question 3. You need to diagnose a suspected cytomegalovirus infection quickly. Standard cell culture takes weeks. Which specific culture technique utilizes centrifugation to physically enhance the speed of viral detection?
The answer is shell vial culture. Centrifuging the sample at low speeds forces the virus into the cell monolayer, bypassing the slow attachment phase and allowing detection of viral antigens in just 24 to 48 hours. This is heavily detailed in the viral isolation section.
Question 4. A patient presents with an acute respiratory illness. A culture of their throat swab shows no obvious cytopathic effect, but when guinea pig red blood cells are added to the tube, they stick tightly to the cell monolayer. What is this phenomenon called?
The answer is hemosorption. This indicates infection by viruses like influenza, parainfluenza, or mumps, which express hemagglutinin on the surface of the infected host cells, causing the red blood cells to stick. Note this is distinct from hemagglutination. Review the detection culture section.
Question 5. To definitively confirm a recent acute viral infection using only IgG serology, what specific fold increase in antibody titer is required between the acute and convalescent serum samples?
The answer is a 4-fold rise. For example, the titer must jump from 1 to 4 up to at least 1 to 16. A smaller rise is not considered diagnostic of a current acute infection. This is a core concept in the serology section.
Final question, question 6. Standard PCR is highly effective for amplifying the genome of HSV. However, which modified molecular technique is strictly required to amplify the genome of an RNA virus like HIV or influenza?
The answer is reverse transcriptase PCR, or RT-PCR. Standard PCR enzymes only copy DNA. You must first use reverse transcriptase to convert the viral RNA into complementary DNA before amplification can occur. Check the genetic detection section for the mechanics.
And that is it for this deep dive into the laboratory diagnosis of viral diseases. Remember, it is an invisible forensic investigation, but if you know what clues to look for, you can crack the case.
Keep looking for those fingerprints.
We'll see you next time.